Good morning, my dear students. Today we are learning about the different type of envelopes present in the cells. So, as you know very well that we have been studied about the definition of a cell, the type of cells, on the basis of a presence and absence of a nuclear membrane, that is a prokaryote cell and a eukaryote cell. You have studied the detailed structure of a prokaryote cell. At the same time, you have studied the detailed study of a eukaryote cell. In that eukaryotic cell, two different type of cells you have been studied, that is a plant cell as well as a animal cell. So now we want to go for start studying all the different parts present in a generalized cell, right? All the parts and their functions. That is the importance of this chapter, because when you will completely know about the cell, then you will get what actually taking place in the tissue, then what actually taking place in the organ, then organ system, right? ನಾವಿವಾಗ ಉಸಿರಾಟ ಕ್ರಿಯೆ ನಡೀತಾ ಇದೆ. ಉಸಿರಾಟ ಕ್ರಿಯೆಯಲ್ಲಿ ಮೂಗಿದೆ, nostrils, that is what ಮೂಗಿನ ಹೊಳೆ, then nasal chamber, then pharynx, then larynx, then trachea, then bronchioles, then lungs. Inside the lungs, alveoli, inside the alveoli the cells will be there, and like that the organ system has been formed, that is the respiratory system. Imagine if any one cell among that will not work properly, then infection will take place, respiratory disorders, it may be cold or a cough, right? So to understand every concept, first of all we must know every bit of the cell. So the envelope comes, in envelope two envelopes were there, one the cell wall and another the cell membrane. So cell wall is present in protistans, 50% of the 50-50. Some of the protistans contain the cell wall, some of the protistans do not contain the cell wall. But fungi, all the cells present in the fungi are consist of cell wall. Then plantae, all the cells present in the kingdom plantae are consisting of cell wall. Then cell membrane present in all the cells, whether it may be prokaryote or it may be eukaryote, any cell. So because of that, first we are learning about cell membrane. So cell membrane, the another name of the cell membrane is plasma membrane. So cell membrane or plasma membrane is outer body covering of the cell. Outer body covers the cell. Cellina ಹೊರ ಪದಿರು, ಹೊರ ಕವಚ ಅಂತ ಹೇಳ್ಬೋದು, ಪದಿರು ಅಂತ ಹೇಳ್ಬೋದು, ಪೊರೆ ಅಂತ ಹೇಳ್ಬೋದು. So outer border of the cell, that is the cell membrane. It is a very thin membrane. Before 1972 or before 1952, when there were no any microscopes like an electron microscope, phase contrasting microscope, stem contrasting microscopes, only generalized microscope was there, who have been discovered by Anton von Leeuwenhoek, and through the help of that they have been started studying the cell. During that time, by taking the cell of a human erythrocyte means RBC, red blood corpuscles. Red blood corpuscles are also biologically called as the erythrocytes, that have a concave shape cell, and that is covered with the help of a plasma membrane. In that they have been started discovered. In that 40% of lipid is available and 52% of a protein is found anta ಹೇಳಿದ್ರು. It is not in all the cell. Remember, very important point it is. 40% of lipids, 52% of a protein is present in the plasma membrane of erythrocyte. Neat level question, right? Whereas species specificity, organ specificity, cell specificity, availability of lipid and fat is different. Sorry, protein. Say for example skin, fat is more compared to the protein, right? So that is why differences. Heart, you know very well, cardiac muscles, the lipids are less compared to the protein. The collagen proteins are more, that is why contract relax. So the organ dependent availability of a protein and fat along with the carbohydrate, minerals, nutrients, right? So the composition of the cell membrane or plasma membrane in erythrocytes or RBC is consist of protein of 52% and lipid of 40%. And however, it is going to be performing the different function, allowing the material in and sending the material out, which it does not need. That we will later study under the function. So like this, the plasma membrane has been given. So late in the year 1972, two scientists called as Singer and Nicholson. Singer and Nicholson are the two scientists has been worked in the year 1972 when the electron microscope was being developed. Through that they come into the conclusion that the plasma membrane consists of a double membrane or a double membrane bound cell. So it is going to be consisting of such a structure. So according to the Singer and Nicholson in the year 1972, the cell membrane they have termed as fluid mosaic model. Most important question for 5 mark for your final examination. Right? Fluid mosaic model of a plasma membrane, or the question asked that explain Singer and Nicholson's model of a plasma membrane, or explain the fluid mosaic model of a plasma membrane. You have to draw the diagram and you have to write the name of the scientist along with the year, then explanation. So let us study. This fluid mosaic model consists of two ball-like structures. This is one layer, this is another layer. So this structure is one layer and this is the another layer. The ball, which we consider them as head, so we call it as a polar head, and this is purely made up of lipid, right? Lipid. Whereas this is also a tail which we are going to be flapping like this. They can flap like this. Understand? They allow the movement in and throw out through the help of these. Along with these channels, I will tell you now. Right? So these are said to be as a tail, called as a polar tail. So hydrophilic polar tail. Hydrophilic means they love the water. Hydrophobic polar head. So polar head is present outside the body, outside side of the cell, external side, not internal side. Consider that this is the cell membrane. Here heads are present here, not inner side. The plasma membrane, when you cut and open, it looks in this way. Right? So polar head and polar tail. So this is one and this is the one. So this is considered as one layer. So this is one layer. This polar tail is also made up of the lipid. Polar head is also made up of the lipid. This complete structure is made up of the lipid. This is the another lipid. So two lipid layers were there. So it is called as a bilipid layer. So according to Singer and Nicholson, fluid mosaic model of a plasma membrane is made up of the two lipid layers. So bilipid layer, polar head and polar tail. Right? And in between that there will be the type of a protein. This is a protein. Such protein which is attached to the both poles, both polar heads. Such type of a protein is called as an integral protein. Integral protein. So here, along with this integral protein, you can go for look over here. Now we will ತೋರಿಸಬೇಕಾಗ್ತದೆ ನಿಮಗೆ ಹೀಗಿರುತ್ತೆ ಒಳಗಡೆ ಹೀಗೆ. Right? ಇದನ್ನ ನಾವು ತೋರಿಸಂಗಿಲ್ಲ, ಅದನ್ನ ಹೀಗೆ ಅಡಗಿಸಿಬಿಟ್ಟಿರ್ತಾವೆ. So consider ಇದನ್ನ ನಾವು ಹೀಗೆ ತೋರಿಸ್ತಾವೆ. It can close, it can open. It can close, it can open. When it open, it can take entry in. When it close, it will not allow to take in. When it open, it is allowing to take in. And it is going to become open to throw it out. Understand? So here we consider it as channels. So protein channels will be present which opens and closes. So they are called as integral proteins. So integral proteins are present. in the plasma membrane. And apart from that, some of the proteins are present like this. Here, outer border layer like this. Like this. Such a type of protein is called as an extrinsic protein. They are called as extrinsic protein. Right? And here, bubble-like structure, a chain of a ball, that is called as a sugar. Sugar molecules will be present. When a sugar considered as a carbohydrate, any carbohydrate it may be, okay? Right? When this sugar is attached to the lipid, we call it as a glycolipid. It is a lipid, sugar is glycogen. So glycolipid, like the same, this is a protein. A protein is attached to the polar head. So we call it as a glycoprotein. Right? So in this way, glycolipid and the glycoprotein are going to be considered here along with the attachment of that. So this is considered to be as a fluid mosaic model of a plasma membrane. Let me to tell you that fluid mosaic model of a plasma membrane was discovered by a scientist Singer and Nicholson in the year 1972. Point number one. Point number two, the plasma membrane is made up of with the two lipid layer called as a bilipid layer. Point number three, the lipid layer consists of a polar head as well as a polar tail. Point number four, there is a presence of an integral protein which is attached for both the border of the polar head. Point number five, here and there is a protein which is present outside of the membrane attached to the lipid called as an extrinsic protein. And to this protein and lipid there is an attachment of a protein as well as a sugar. Right? This is the structure of a fluid mosaic. Then what is its functions? Let us see. As you know very well that plasma membrane is allowing the material to take in and to send out. So it is acting as semi-permeable membrane. Point number one, function. It is the most important. So functions of a plasma membrane, it acts as a semi-permeable membrane. Why we call it as a semi-permeable? Semi means it is selectively takes whatever the cell is in need and it will not take what it do not need. Understand? This is an important. ಇವಾಗ ಒಂದು ಕೆಲಸ ಮಾಡೋಣ, ಏನು ಕೆಲಸ ಮಾಡೋಣ ಅಂತಂದ್ರೆ ನಿಮಗೆ ಸಮ್ಮರ್ ಸೀಸನ್ದಲ್ಲಿ ಮೇ ಮಂತದಲ್ಲಿ ನಮ್ಮ ಕಾಲೇಜಿದ ಟೆರೆಸ್ ಮೇಲ್ಗಡೆ ಒಂದೆರಡು ಗಂಟೆ ನಿಂದ್ರೆ ಸರ್ 12 ರಿಂದ 2 ಗಂಟೆವರೆಗೂ ನೀರು ಕುಡಿಸಂಗಿಲ್ಲ, ಏನಿಲ್ಲ. ಏನಾಗ್ತದೆ, ನೀವು ನೀರೆಲ್ಲ ಹೋಗ್ಬಿಡೋದು. ಅವಾಗ ನಿನ್ಗೆ ಒಳಗೆ ಕುಡಿಸಿ ಕ್ಲಾಸ್ ರೂಮಲ್ಲಿ ಕುಡಿಸಿ ನೀರು ಕುಡಿಸಿದ್ರೆ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ನೀರನ್ನು ಹಿರ್ಕೋತದೆ ಹಿರ್ಕೊಡೋದು. ಆಲ್ ಸೆಲ್ಸ್ ವಿಲ್ ಅಬ್ಸಾರ್ಬ್ ಬೈ ದೇರ್ ಓನ್. ಅವಾಗ ಅವುಗಳಿಗೆ ಎನರ್ಜಿ ಬೇಕಾಗಲ್ಲ. Understand? Because they will take such type of an absorption or making the entry of a fluid inside the cell without spending any energy. It is called as a passive transport. Understand? It is called as a passive transport. Then another active transport is nothing but consider here outside the cell membrane. Consider this is in a cell and around this cell the blood vessel is going to be running and from this blood vessel the blood cells will be present here along with the minerals will be there. Consider this is in need of a sodium as well as a chloride ion. This cell is containing less amount of chloride ion as well as sodium ion, but here sodium as well as the chloride ion are more. Now movement of a molecule from a higher concentration level to the lower concentration level, higher concentration level to the lower concentration level through the help of a semi-permeable membrane called as osmosis. So osmosis is going to be taking place through the help of the cell membrane. You have been studied the second function, right? And osmosis you know. And without any spending energy, when the material taking in, that is called as a diffusion. In detail, in plant physiology, you will learn these all the concept. One the period ಬೇಕಾಗ್ತದೆ osmosis ತಿಳಿಸಾಕ ಒಂದು period, diffusion ಮತ್ತೆ imbibition ತಿಳಿಸಾಕ ಒಂದು period ಬೇಕಾಗ್ತದೆ. You will learn there. Just you have to feed from a region of a higher concentration level to the region of lower concentration level through the help of a plasma membrane is called as osmosis. Without spending any energy, that is diffusion. That's it. Along with that, we consider endocytosis. Endocytosis. Endo means to take in. It is going to be taking in. If mineral is taking in, endocytosis. If water is taking in, it is also endocytosis. But when it is a molecule, mineral, it is not drinking, it is eating. So cell is going to be eating and water is not eatable. Cell has to be drink it. So we call it as cell drinking. So endocytosis consists of two phenomena. One is said to be as phagocytosis. Phagocytosis. Phago means to eat. Cytosis means cell. Cell eating. Cell is taking the solid material into the cell through the help of a plasma membrane is called as phagocytosis. Right? Whereas another one is said to be as pinocytosis. Pino means to drink. Cell is going to be drinking. Right? Cell is taking the fluid material inside the cytoplasm through the help of plasma membrane. That is called as a pinocytosis. So this is said to be as an endocytosis. Say for example, WBC is there, white blood corpuscles. So white blood corpuscle is this is the structure and this is the nucleus, and here cell organelle will be there, and this is the cell membrane. And now this is the pathogen. This is causing one. Now this is going to be eating it. What it happens? Phagocytosis or pinocytosis? Cell is eating the solid substance to kill it. Not to kill it only, to utilize it. That is called as phagocytosis. When the plasma membrane is taking the fluid material inside, that is called as pinocytosis. So cell eating, cell drinking, generally we consider that is the. So let me to tell you once again the functions of a plasma membrane. First function is it is a selective semi-permeable membrane. Second one, plasma membrane acts as an osmosis, that is a movement of a molecule from a higher concentration level to the lower potential through the help of a plasma membrane. Diffusion, movement of a molecule from higher level to the lower potential, right, without the energy. And active transport, passive transport. Pinocytosis, phagocytosis, considered to be an endocytosis. When cell is taking all the fluid material, everything taken in, the cell is going to be generating waste substance. That waste is throwing out from the cell, throwing out the waste from the cell. That is called as exocytosis. What is it called as? Exo, throw it out through the help of the cell. Right? So these are the functions of plasma membrane. I hope you have been understood. In the next week, we will go for studying about cell wall. Thank you. Thank you.